Well, welcome everybody to our February conservation chat. Julie Bates could not be with us today, so I'm taking her role. Uh, Thank you again for joining us. And please tell your friends about how wonderful these conservation chats are. And be sure to join us for our monthly chat, which we'll talk about more at the end of this discussion. I'm Deborah Olson. I'm the Executive Director of the International Elephant Foundation. And I would like to introduce to you Sarah Conley, who is our Director of Conservation, and she can take it from here.
Thank you, Debbie. We have a wonderful program for you guys today. We're very excited. We've been planning this one for a while now. Today we are joined by Stacey Engel. She's a PhD she's a PhD candidate in molecular biology at Stellenbosch University in South Africa. Her work aims to better understand the immune responses of free-ranging African savanna elephants to infectious diseases such as tuberculosis. As part of her work, she's assembling a trans transcriptome. I'm sure we'll learn more about that and how to pronounce that word when she talks today for African elephants to facilitate future gene expression analysis and disease biomarker discovery. Stacey is working with Dr. Michelle Miller, who's head of the Animal TB Research Group at Stellenbosch University. And Dr. Miller has worked with elephants for a long distinguished career, including as the veterinarian for the elephant tag here in the United States. Dr. Miller has a long relationship with IEF, and we've been working with Stacey for a while as well, and we are so pleased to have them both here. Stacey, welcome. Thank you for being here and doing this.
Hi, everyone. Thank you, Sarah, for that wonderful introduction. I'll just get my presentations going.
And while Stacey gets everything ready, a couple reminders for the audience. If everyone can please keep their cameras and sound off while we're doing the presentation, and that will help us conserve bandwidth for our international attendees where Wi-Fi and connectivity might be a little more difficult. So thank you. All right, Stacey, take it away.
I just want to check, Sarah, you guys can see my screen?
Absolutely.
Yeah, perfect. So, good day, all. I'm Stacey Engel, a PhD student from Stellenbosch University in South Africa. And I work on one of the projects funded by IEF in 2024. As a brief background, our research group focuses on tuberculosis, which is a bacterial disease of the lungs that affects humans and animals. And TB is still a major problem in South Africa, affecting humans, livestock, and wildlife, including elephants. And since elephants are long-lived, they can potentially be infected for years. And therefore, we wanted to understand how these animals actually cope with the disease. Today, I will cover some of the interesting things we have learned, as well as the challenges of this project. I'd also like to add a little disclaimer that all of the pictures of elephants in recombinant position in this presentation, they were all visibly healthy and are under chemical immobilization under the supervision of a senior vet. Just getting my little pointer ready. So Kruger National Park is well known for its biodiversity and the opportunity for visitors to see the big five, including elephants. The park's elephant population is estimated to be approximately 31,000 and is one of the largest on the African continent. Although tuberculosis is present in humans, livestock, and some wildlife in South Africa, it was never really considered a threat to the elephant population. Although this all changed in 2016, when our research group leader and the Sandpark senior veterinarian investigated a case of an adult bull that had just died. Often, older elephants die from worn out teeth or sometimes fights with other bulls. But this animal did not have any signs of trauma nor worn out teeth. Yet, he was visibly thin, suggesting a chronic problem. Upon examination of the elephant, they found that he had died of severe infection in his lungs and other organs, which appeared to be tuberculosis. Surprisingly, this turned out to be the first case of human TB in a wild African elephant that had no known contact with people. since diagnostic tests for tuberculosis often depend on the immune response and are very limited in elephants. Our first step was to gain a greater understanding of how the elephant immune system functions. Since then, TB has been documented in African elephants in Kruger National Park, Tsavo National Park in Kenya, a zoo in South Africa, and zoos all around the world. A pilot study screening Kruger elephants suggested that approximately 1 in 12 elephants could actually be infected with TB bacteria. TB in elephants appears to mainly affect the lungs and the lymph nodes, similar to humans. However, like humans, only some elephants actually develop the disease, resulting in weakness and weight loss, although it can take years or even up to decades to advance to a point where these signs are actually seen. If we better understand how the elephant's immune system copes with the bacteria, we'll be able to improve the tests for detecting infection early and even prevent spread to other human and animals. This is important because TB doesn't only impact individual health, but it's also a threat to elephant populations if infected animals are moved for conservation programs, and especially because African elephants are currently considered an endangered species. So this then led to my current project, which is to study how the elephant's immune system works using immune cells from the blood and the lungs. By then identifying the genetic messages from these cells, we can then create a molecular library of information that can be used to improve diagnostic tests for TB, as well as other important diseases. In order to find these molecular pathways of how these immune cells function, first, we needed to be able to create a system to challenge these cells in our laboratory. Then by looking at the messages that is associated with the activation of these cells. In the next step, we wanted to compare these messages that occur in the blood cells to those that occur in the lung cells. Finally, we then plan to use this information to create tests that allow us to easily detect when an elephant's immune system is responding to different diseases like TB. Of course, it is easy to get a blood sample from a person, but imagine having to take a sample from a wild elephant. And this requires a lot of expertise, and of course even luck, to find an elephant to test, safely administer the drug for it to fall asleep, and then handle the elephant. Fortunately, we have a professional wildlife veterinary team to help us do this, which even requires a helicopter for the vet to administer the drugs via a dart in the rump. Once the elephant is in laying down, the team can then safely approach the elephant and ensure that it is doing okay. Then only we have the opportunity to collect our samples. So for our project, we collect blood and respiratory samples from the lungs as well as the trunk. In addition to our research samples, there are also other samples that are collected, and they are preserved in almost a frozen zoo to help other current and future research on elephants. And this is to maximize the contributions that each elephant makes to science. So as I've mentioned, we really want to understand what happens in the lungs as well as in the blood. Therefore, our team has developed a unique system for safely collecting samples from inside the lungs of the elephant. First, we put this tube into the airways, which is this blue tube in the picture, into the airways of the elephant, and then insert a custom designed endoscope, which is basically a long, flexible, drivable tube that has a camera attached. And we insert this down into the smallest airways. And this is similar to playing a video game, as we can then see the inside of the lungs on the screen as we proceed deeper and deeper into the airways. We then put sterile fluid into the lungs and suction this back up into a sterile container to collect any bacteria and cells that were in the lungs. This procedure is very safe and routinely done in humans and domestic animals, just not as common in animals the size of an elephant. On the left is a picture of our sampling equipment, which is 3 meters long, or approximately 9 feet long, to reach into the elephant's lungs. On the top right picture, you can see the screen where we can see where we are in the lungs and use this to sample different areas in the lungs. The lower right picture shows the collected lung fluid in a container that will go back to the laboratory for processing. Our first challenge was then to figure out how to recover these immune cells from the lung fluid. And this is the first known study to use these techniques in elephants, especially wild ones. However, nothing is ever as easy as it seems when working with elephants. And we initially thought that using techniques based on humans and other animals would, of course, be successful in elephants. But what we found is that because elephants use their trunks for everything from exploring the environment, eating, drinking, and greeting other elephants, this affects what goes into the airways. And in order to deal with the lots of inhaled material, they appear to produce this really thick mucus, which helps to trap debris. But it also interferes with recovering the immune cells from the samples. And sometimes we found dirt, ants, and grass in the samples, as you can see from how dirty this lung fluid is. And therefore, we had to figure out how to overcome these challenges. And this actually took much longer than what we originally anticipated, delaying progress in our project. However, we then finally came up with this multi-step method to successfully get the samples we need to move forward in our study. And this is also in light of having to perform sample processing in a field laboratory for our initial sample preparations. After lots of trial and error, we were then finally able to successfully recover the immune cells from the lung fluid. The microscopic images of cells were examined by a specialist vet that could help us evaluate and characterize these different cell types. These first ever results were published in a scientific journal so that other vets and research scientists could learn from our efforts. After overcoming the hurdle of recovering the cells, we now needed to focus on how we could actually activate these cells in the lab in order to study those immune function molecular pathways. And after testing various conditions, we were able to optimize the method to activate both blood and the lung cells. And we found that one stimulant in particular worked the best. So we used these same conditions for all of our samples, including the blood and the lung cells. And this allowed us to watch how the cells react when they think they are fighting a disease, and then to see the messages that they send to each other during this immune response. Of course, science doesn't always go that smoothly. And after stimulating the cells, we noticed something unexpected. The blood started to clot when we tried to preserve these samples, as you can see in these pictures. And instead of a nice clean samples, we now had these clumps forming. This made it much harder to recover the molecular message, indicating an immune response. And what made it even more surprising is that we've never actually seen this happen in any other species before. So these elephants definitely continued to challenge us. Finally, we had a breakthrough. After a lot of troubleshooting, we were able to determine which genes were switched on during immune responses. And this allowed us to trace step by step how the cells were mounting an immune response at the molecular level. But before we could actually fully understand these messages, we first had to build a complete library to interpret the genetic messages being sent. And that's where the next challenge began. Because building a genetic library for a species that hasn't been fully mapped is no small task. So the problem was that we needed to sort the messages, putting the information into the right sequence to make sense. But eventually, after refining our approach, we were able to untangle those messages and build a working genetic library for the cells. And this library was assembled with the help of highly specialized bioinformatics software. And this is where it gets really exciting. So we can now use this information to search for special signals in the blood. And so these markers that can tell us when an elephant's immune system has been activated. And that brings us closer to our ultimate goal of creating a simple test that can detect immune activation in elephants. So what is the purpose of all this? What is the point of actually doing this project to understand the immune response of African elephants? Well, since we still know surprisingly very little about how the African elephant's immune system actually works, especially at the level of individual cells and the messages they send, this project aims to answer a lot of these unknowns. Furthermore, we are also building new knowledge that doesn't just stay in the lab, but it can help shape better diagnostic tools and improve how we monitor elephant health. We are also creating a toolbox for vets and researchers that helps them detect disease earlier and manage elephant health more effectively. It may even help us understand how the elephant immune system differ from our own. Our results will also support future research and help identify disease markers and deepen our understanding of elephant immunity for years to come. And then to end it all off, all of the investigators on this project would really like to thank the IF for all the continued support and the funding, which allowed us to do all of this research and work together for the benefit of these elephants. Thank you all for listening, and I've also included my email address if anyone would like to discuss anything further or if you'd like to collaborate on anything. Thank you all.
Thank you, Stacey. That was fascinating. I know a lot of people are going to just love to know your process. But because we're talking to a mixture of scientists and lay people and conservationists, can you walk us through the logistics and maybe number of personnel and how you go through planning these immobilization operations and then kind of maybe a typical day accomplishing one of them?
That is a great question on the planning of it. I'm sure Michelle and Tanya can agree. It's definitely no easy task. It takes us probably like 3-4 months to actually get the project planned. And since we do a lot of our research is based, or a lot of the initial lab work and the cell isolations happens in Kruger National Park, it requires us to do all the planning, order everything we need from every little test tube, every little piece of equipment to actually then transport that over to Kruger before we can even start the sampling. Then there's also the logistics of organizing the helicopter, the wildlife team as well, and some of the members of that team is actually in this picture over here, and the rest are all members from Stellenbosch University. So it really does take a whole team to actually sample these elephants. And then typically how a day would go. So we go about once. I think we've been for about 3 sampling opportunities since we started this project in 2023. And typically we would start really early, about 5-6 o'clock in the morning, and we'd head out to first find an elephant to immobilize. And once the elephant's immobilized, like I said, then the ground team goes ahead and make sure the elephant's okay, and we would then begin sampling. And that would take us to about late morning, and we would basically spend the rest of the day in the lab trying to isolate those cells from the lung fluid. So it is a very long day with a lot of planning indeed, but it's definitely all worth it.
Fascinating. And so you need to process those samples right away, same day.
Yes. It's because we want the cells alive so that we can actually activate them. So we try and do that as soon as possible in the lab.
Wonderful. Um, just a side note, in your presentation, you talked about how you use a special endoscope to go and collect the sample. I know from elephants in human care that they have lost endoscopes, but they weren't sedated at the time. So the elephant sort of smushed it not in their mouth, but in their throat. Have you ever had that happen with any elephants you've been sampled, or does the sedation get rid of that problem?
That is actually quite interesting. No, we haven't had that issue. And I think that's because we use this little the blue tube that I was showing in the picture. So that's an endotracheal tube, and that kind of takes us from the mouth through the trachea of the elephant. So that kind of causes a little shield around the endoscope. Because absolutely, these endoscopes are really fragile. So I can imagine how they would easily be able to crush this tube.
Yeah, and that would add significant cost to an already expensive operation, I'm sure.
Yeah.
Just a reminder to everyone listening, please put your questions in the chat. We would love to know what you're thinking about and what your questions are for Stacey. In the meantime, why don't you tell us about what your next step would be? What are you working on next, or what are your sort of longitudinal plans for this research?
So for now, we've just finished developing this transcriptome, or in my presentation, I've called it a genetic library, just so it's a little bit more easily understood by everyone in the audience. So we've just completed that, and that is looking really good. And the next step is then to just kind of tease out all of this messages and try and find out which one actually shows that these elephants are infected with TB or where they've been infected with different diseases like EHV as an example. And then to basically just use that to get more specific tests to diagnose these diseases. So that is our ultimate goal, and hopefully we can get closer to that goal in the next year or two. For now, we're just trying to get some more elephants in our on our sample list that have actually been infected with TB.
Of course, yeah. So as you were immobilizing wild elephants, did you find any that had TB?
Since I've joined the group and the ones that we've immobilized, we have found one that had trace TB detected.
Wow,
And there've been some others, but unfortunately not the ones that we've sampled in our project.
sure. Um, and have you actually pulled blood from an elephant to see if it is mounting an immune response to this? They have developed this process, so has it been tested in real time?
Yes. So we do collect blood to do those tests, but unfortunately we don't use those blood tests to diagnose TB. We do cultures, but absolutely, yeah, we do that.
Awesome. All right, a more technical question. Is the cell activation primarily being looked at in the macrophage line, lymphocytes, or other? Are there specific markers, proteins that indicate the cell activation?
So for the blood, we are just looking at the blood as a whole because we're trying to get a peripheral circulation response. And then for the lung cells that we are looking at predominantly alveolar macrophages. But when we isolate the cells, we don't specifically select for macrophages. We kind of do a gradient separation. So that will include some neutrophils, some other granulocytes. So, but it is predominantly alveolar macrophages. And then we don't have specific biomarkers or proteins that we are looking at. So we're trying to kind of take it from a much broader approach. And that's how we're kind of doing this transcriptome or the whole genetic library approach. So taking a step back and now seeing what's actually increased or decreased when the cells are activated, and then looking at those specifically.
Wow, awesome. Uh, okay, we have a more personal question. What is your background? Do you have professional training? How did your passion for research and elephants conservation start? People are always interested in how our experts in the field got to where they are.
Oh, that is a cool question. So I actually only started working on elephants in my PhD. In my master's and honors work, I was looking at a specific protein in in TB infected in humans. And then for a PhD, I got this really great opportunity to then work with the animal TB research group. I thought it was an amazing opportunity, obviously, to work with the elephants. I mean, who can say no to such an amazing opportunity? And then since starting my PhD with this research group, I've kind of just gotten training on the go, I guess. I literally registered for my PhD the Friday and the Monday I was in Kruger collecting samples. So it's been a great journey.
Wow, that's awesome. Um, as a kid, were you in love with wildlife, or was this something that sort of intellectual curiosity brought you toward?
I would say a little bit of both. Um, I definitely have always been interested in wildlife and particularly elephants, but I would probably intellectual curiosity is what also got me a lot closer to actually choosing this project. I wasn't initially planning to do a PhD. And I thought the opportunity and the skills that we could learn and the things that we could do for elephants with this project would have been amazing and definitely not an opportunity to miss. So a little bit of both.
Wonderful. Uh, you mentioned that there is a way or possible implications that this can tie to EHV. Maybe tell us a little bit more about what you're thinking with that.
Yeah, so similar to with the TB, how we are hoping to eventually get a diagnostic for TB, we are hoping to do the same for EHV. So once we get the elephants that are infected with EHV, we can then find what are some of the messages that are specific to EHV infections. And we can then use that to build more specific tests or more sensitive tests to diagnose EHV in these elephants.
That would be wonderful. Um, and when it comes to TB, can you grow that immune response cells? Can you grow those cells in the lab? And if so, uh, if you can grow those immune cells in the lab, would that potentially have applicability for a treatment or to boost an animal who is ill, some sort of way to help their immune system?
So I assume it's similar to what happens with humans. So we can grow the TB bacteria in the lab. And I guess it's similar to what happens in humans in South Africa. When we're born, we get a BCG vaccination, which is kind of like attenuated TB strain. Um, so, I mean, that would also be a good thing to be able to do that in elephants is to get a vaccine for TB so that the elephants can really start building an immune response against TB. So, yeah, that is definitely something in the works as well.
Absolutely. Okay. We have a multi-parter here. So if we need to circle back, just let me know. If you had an idea, do you have an idea why the blood coagulated in your goal one experiment? And could you expand a bit more on how you correlate immune cells, immune messages to the diagnosis of TB in elephants, perhaps related to the fact that symptoms can take years to develop after infection?
So the first question was on the blood clotting.
Yes.
Um, so to be honest, we don't know an exact answer. We have a little bit of theories. It could be some of just the blood cell composition of elephants compared to other animals. So they've got a larger portion of macrophages or monocytes in their blood. It could just be some of the higher fatty content. That was something we were exploring as well. And then even just exploring, could it just be mucus in there as well? So I spent some time optimizing that, trying different ways to kind of break down those clots. But unfortunately, it wasn't successful or viable to go that route, and then we just ended up getting a different way to preserve these samples. But yeah, we don't have an exact answer of what exactly that was. But it's really interesting that we only saw it in elephants. For example, like we haven't seen it in rhinos, buffaloes, or some of the wildcats that we've sampled. And then can you just repeat the second question, Soda?
Absolutely. That was really interesting. Uh, they're asking, could you expand more on how you correlate immune cells or immune messages to the diagnosis of TB in elephants, especially the delayed onset of symptoms?
So the delayed onset of symptoms um, doesn't necessarily correlate to those messages. We're hoping that the messages could be sent a lot earlier. So what we're looking at specifically is the RNA, which is just basically what is expressed in these cells at a specific time. So the theory is that when these cells are exposed to a certain bacteria or to a stimulus, is that they would then express certain RNA or certain messages. And we're hoping with that, we could then be able to look at that response and those patterns and be able to determine whether the animal might have TB or not, rather than waiting those years or even decades sometimes for those signs to actually show. Because I mean, by then you can imagine that elephants traveled quite a bit. might have been translocated, and you can imagine all the other elephants he's actually infected along the way.
Great point. Um, Someone's asking if there are any less invasive methods of detecting TB or the immune cells for TB, like feces, things like that, since obviously these darting operations are pretty costly and invasive.
That is a great question. So there has been traces of TB found in feces of elephants, but because of shedding, you can't always trust that method of detecting TB. So it might be that sometimes bacteria is found in the same elephant today, but maybe in three days' time, the bacteria might not be present, and that's just due to shedding of the bacteria. But it doesn't mean that the elephant doesn't have TB 3 days later. So the current gold standard is a trunk wash. So we form a trunk wash and then to do a culture of that. And obviously in zoos and working elephants, it's a little bit easier. Some of the elephants are actually trained to do this. It just becomes a little trickier when working with more free-ranging elephants that aren't trained to do it. But yeah, it would be great if we could diagnose it using feces, but I don't know. I don't know who you'd have to convince to get that as an improved test anytime soon.
Well, it sounds like it's unreliable for now, so can't be used to make important decisions like that. Well, so say you found a wild elephant while immobilizing, or maybe not, that you knew had TB. Would you treat it, or how would you treat it? Would you mark that elephant so everyone would know, so you wouldn't necessarily translocate it, like you mentioned earlier? Um, just a hypothetical.
So the tricky thing about these sampling is that we sample them while we're in Kruger, but we don't do the test for TB until we get back to Stellenbosch University. And then it takes about 52 days for the TB to actually grow. So realistically, we only know in about 3 months or 2 to 3 months whether that elephant actually had TB. And by then, that elephant's gone on its merry way again in Kruger National Park. So unfortunately, it's not that easy. Also in South Africa, for free-ranging African elephants, we don't treat them.
Okay, that's good to know. Um, and when you're selecting an elephant to immobilize, how do you do that? Is it opportunistic, or are there specific characteristics of an animal that you're looking for? Maybe walk us through that process.
So that's a little bit of both. So it is opportunistic, where if we find an elephant, that's obviously one that we prefer. And we do have a specific area in Kruger National Park that we kind of sample within. And then, like I said, we try and look for these lone adults. And usually the vet's the one who's in the helicopter and administers the drug. And then he will kind of give an okay of whether the elephant is an adult, whether he's old enough, whether he looks healthy, and he will give the final say. So a little bit of both, that we do kind of make sure that he looks healthy and he's not super old or super young. But again, it depends on who we find in that area.
Sure. And about how many collaborators, personnel, people are involved in one of these operations?
Oh, um, I don't know the exact number, but usually it's about the head vet, then maybe the ground team, like about 4 or 5, people, because they've each got their different roles in the ground team, and then we've got our own Stellenbosch team that also assists with the sampling. So probably around 10 at a time, but that's would just be me guessing a number right now, more or less an educated guess.
That's amazing. I mean, it really takes a village to do these types of things. I think people just assume that, it's one person in a lab doing one thing, and it really takes a lot of you.
Mhm. I mean, even in the lab as well, it's not alone. We are not alone in the lab. At any given time, there's probably about 2 or 3 people also working on these samples, just because it's such a lot of work to do, and so many samples to process.
Oh, so true. Going to samples, are you focused just on male elephants, or would you maybe have different classifications for females, especially with hormones and calf rearing?
That is a really interesting question because I mean, in humans, you can see how um, the the TB or the incidence of TB differs between male and female just because of their sex hormones. So I mean, that'll be really cool to see in elephants. But the issue is that the female elephants obviously tend to travel in herd. So to be able to immobilize one elephant at a time, it kind of then stresses out the herd. So we're just trying to avoid that. So we tend to rather do the lone males. So it is a little bit of a skewed project, and that we do focus mainly on males. But I mean, it's better than stressing out the herd for just a sample.
Absolutely. Uh, just a comment from Dr. Miller in the chat, which I think maybe will educate on some things. TB is endemic in wildlife in Kruger National Park. Therefore, these animals are not treated or removed. Susceptible species in Kruger are quarantined. They cannot be moved out of Kruger without testing. This is to prevent spread to other areas. So that sort of answers one of the other questions about treatment and what you would do if you did find someone or an elephant that was positive. Thank you for that, Dr. Miller. Um, let's see, what else should we discuss? This is so fascinating. We have so many people engaged. Um, how do you envision this moving forward? What 10 years down the line, what would you like to see this project accomplish?
I think something that I think is really cool that we could accomplish with this project is that we wouldn't need to do these trunk wash samples or these bowel samples to actually diagnose diseases like TB in elephants. We could use something like blood. And the idea of this project is to find a proxy for what we see in the lungs in the blood. So if we can kind of compare those immune responses and see, hey, we're seeing this increase in the blood, so that means this must be happening in the lungs. So we wouldn't need to have to do these lung fluid washes in the elephants. We wouldn't need to do trunk washes in the elephants. We could just draw blood, which I think is a lot less invasive than doing the bowels and the lung washes and the trunk washes.
That would be incredible. Um, this, I think this coming from someone with elephant management experience. So when you've immobilized an elephant, how do you get the trunk wash? Uh, because obviously, if you have a trained elephant, it blows the saline or whatever out. How do you do that with an elephant that's been sedated?
Um, so it's actually it seems a little silly, but well, the elephant's trunk is kind of just laying out, and then we pour saline liquid down the trunk, and then someone just kind of washes the trunk out. So kind of flows the liquid further in the trunk and then bring it out. And then we use in a vacuum to just suck up all the liquid that we've put in the trunk. And we'll
Oh,
do that for nostrils.
okay. Wow, that's incredible. Very cool. Okay. Um, what kind of considerations do you have when you're immobilizing an elephant? Obviously, there's probably a time factor. How long can they be down like that? What types of things do you monitor?
So we've got the head or the senior vet at Sandbox there that is with us the whole time, monitoring the elephant, monitoring the elephant's breathing, how the elephant is responding to the drug, and then also timing us to make sure that we're not taking unnecessarily long. He also monitors kind of just the general health of the elephant, like whether it's looking really thin, how old he is, and then also when we're doing the lung washes, like I said, we've got the little endoscope with the camera, so we can kind of get an idea of how the lungs are looking as well, whether he's looking sort of healthy, whether there's a lot of bloody spots in the lungs, and we can kind of tell quite a bit from those things, or those things we then consider for the elephant's health.
Yeah, no, very important. Okay, follow up to the trunk wash question. So say the liquid that is sucked out is dirty. Is that liquid actually in the lungs, or do they have a trachea like that humans have that prevents dirt and debris from going into the lungs?
Um, I would definitely think not. I mean, we've seen some crazy things in the lungs. We've seen some twigs in the lungs. We've seen leaves, and this is with the endoscope deep into the lungs. We've seen some twigs, some leaves, and we've actually have this little forcep adapter where we could actually pull out some of these leaves from there. And then yeah, the trunk wash is also really, really dirty. So we just do with what we got, but they do come out really dirty, similar to the bowl.
Wow. Okay. Uh, Question about after when you've kind of rewoken the elephant. Uh, would they feel that something had been done? I mean, what obviously we can't know that and we don't, want to anthropomorphize, but what do you perceive when they've sort of been, when the reversal meds are given, how they react?
That is a really interesting question. Um, so I think in terms of like the trunk wash and the balls, I don't imagine that they feel much just because of the little amount of liquid that we actually fill or use to wash out their lungs, which obviously a much larger size. And I mean, in humans, for bowels, it's humans tend to say it kind of feels like a little irritation, like a light irritation. So I imagine it'd be similar to humans where it doesn't, it's not as bad, I guess, but more like a little irritation. Um, and then how the elephant um feels otherwise, yeah, I'm not sure. But when he wakes up, the first few seconds he does seem a little bit confused, but after that few seconds he just goes about on his way again. So I don't think it's too bad.
Yeah, it doesn't sound like they are bothered too much. Uh, it's just like they had a nap, I guess.
Yeah.
So going back to leaves and things in the lungs, how do you, can you maybe tell us about how elephant lungs are, how they can sort of live and be healthy with that kind of dirt and stuff in their lungs? How are their lungs built differently than ours?
I mean, that's exactly what we're trying to figure out as well. How do they deal with all these different things? I mean, we've even found ants in there as well. As I was processing the cells, I've had to filter out some ants in there as well. And the samples to the end is still really, really dirty, but yet they seem really fine. Yeah, they're still perfectly fine. So that, I mean, that's something that we would also like to answer. And I think apart from that, I think the size of a leaf or a piece of grass or an ant, I mean, compared to the elephant's lung, it must be really, really tiny. I think in a human lung, it would be a little bit more significant. Um, but in an elephant's lung, it probably is so insignificant. But that is definitely an interesting question.
Wow, ants in a lung. That is insane. Okay. And then we always like to ask people about if you have any interesting stories in the field, something that went unexpectedly and you had to pivot, any sort of, I don't want to call them fieldwork fails like you see on social media, but just kind of weird stories that you could share with us to give people an idea or flavor of how things are.
Oh, I think the most interesting field experience I've had was finding those leaves in the lungs. And I kid you not, the whole team was in hovering around this little screen, seeing the leaves in the lung. I mean, no one could believe that was happening. Um, yeah, I don't think I have any much more other stories um, than that. I mean, our field work is quite controlled, so
No,
that as
that's
bad as
good.
it. Yeah, it's actually a good thing for once.
Uh, Out of curiosity, did you determine what plant's leaves they were? What kind of leaf?
I wish. I mean, it came out and just it was covered with like mucus and a whole lot of fluid that we just kind of shoot it to the side.
Sure. Yeah, of course. But wow, amazing. Okay. I think we have mined every question. I don't think I missed any. Um, but what what do you think that the normal person, you're out there in the field, you're doing great work for elephants, but what can a regular person do to help contribute? Um, what people always want to know how to get involved. So maybe maybe tell us from your perspective.
Oh, um, I think it's always good to understand the problem. Um, I think people have this reluctance to get involved, and I don't specifically mean this crowd. I think just in general, people have a reluctance to help out animals, help out wildlife because they think, it's kind of like so segregated that the human life doesn't necessarily depend on the animal life. But I mean, what we found with even disease spreading between the two, where humans have actually gotten TB from elephants, is kind of just to me, it's just kind of just to be more aware of the importance of wildlife and kind of this One Health approach, taking consideration the animals, um, even like the wildlife, livestock, and then even just environments as well. So kind of just trying to understand that it's a more holistic approach. People tend to kind of just focus on the human side of things, when actually it's a lot bigger than that. So I think just trying to get more involved and get a better understanding of that.
Absolutely, I couldn't agree more. We're all a part of nature, so we're all connected, whether we realize it or not. And that separation of humans from nature and humans from elephants and animals is only going to harm us in the end. So,
Absolutely.
um, thank you. That was a excellent answer. Um, well, at this point, we're going to wrap up. So, if everyone wants to unmute and turn their camera on, we can give Stacey a big round of applause. This was fantastic, and I know everyone learned a lot. Big applause for Stacey. Awesome. Thank you so much.
Thank you, Owen. Thank you for the opportunity to share my work with you guys today.
Well, it is our pleasure to connect people like you to our followers and spread great information out there. So thank you. I'm going to turn it over to Debbie, who I think has some closing comments.
Yeah, I just want to say that for everyone who listened, if you're interested in supporting projects such as Stacy's, um, or any other of the elephant conservation research projects that the International Elephant Foundation funds, please visit our website and consider making a donation. We would greatly appreciate it. And stay in contact with us. We always like to hear from people who are supportive and wanting to learn more about conservation.